Good afternoon or good morning, wherever you are. It's my pleasure to welcome you to the Alice and C.C. Wang Molecular Parasitology Award Symposium. I'm Patricia Johnson, and I'm this year's recipient of this award. Um, I want to start by saying a few words about Alice and CC. As you can see in this picture to the top left, this is a picture of the two of them at the Great Pyramids of Giza, just right outside Cairo, on one of their many trips around the world. They were partners in both life and in science. And when they were not traveling the world, they worked together as a scientific team for many decades at UCSF. And there, Alice and Cece established many molecular biology techniques, which opened the door for mechanistic studies on several neglected protozoan parasites, including Trichomonas vaginalis, which is the organism I work on, and I'm going to talk about today. And in fact, without Alice and CC, it would have been much more difficult for me to change organisms at the beginning of my career. I switched from one parasite to the other, choosing Trichomonas vaginalis, and they helped me in every way possible, being the only two people in the world who actually did any molecular biology on Trichomonas at the time. I'll always be so grateful to them. But they also, in addition to their work on Trichomonas, worked on Giardia and the African trypanosome. They made many insightful and seminal discoveries on various topics in various of these organisms, including nucleotide salvaging pathways, looking at specialized organelle biochemistry, and were in fact the discoverers of double-strand RNA viruses and protozoan parasites. So the two of them were among very few founders in the fields of biochemical and molecular parasitology. Unfortunately, we lost CC in 2017. We all miss him dearly. I want to just point this out, the picture here on the slide of CC sitting in front of the tissue culture hood. You see him smiling. I think one of the reasons he was smiling is he loves science. He loved doing science. He was also, just as a person, someone with such a warm heart, this huge smile, often from ear to ear, and a booming voice. If CC was around, you usually knew it. You could hear him. But what really also was a real feature of him was his generosity and his curiosity. And through this, he was a driving force in creating and unifying the community of scientists that are currently studying molecular parasitology. So both Alice and Cece were very devoted to building the field of molecular parasitology, terrific mentors to many junior scientists, including myself. So I'm actually humbled to have received this award in their name. So now I want to really quickly go through the speakers that we have in the symposia today. As I've already introduced myself, I'll be speaking first. I'm from the University of California, Los Angeles. Our next speaker will be Bill Petrie, who's at the University of Virginia, and he'll be talking about amoebic colitis, the interaction of the microbiome, the mucosa immune system, and the parasite. And then Steve Beverly will follow him, talking from Washington University School of Medicine. And Steve will be talking about trapped persistent RNA viruses as virulence factors in protozoan parasites. And then Mimi Ho, our junior faculty, will wrap up the symposia. She's at Columbia University, and she'll be talking about native structure of the Plasmodium falciparum ROP-H complex determined using an endogenous structural proteomics approach. So can everyone hear me just fine? I forgot to put my earphones in, but can someone speak? Should I maybe, no, I can't hear anyone. Technical staff?
Yes.
Pardon? Okay, all right. So All right, I will continue then. So the title of my talk is Interactions Between the Sexually Transmitted Parasite Trichomonas vaginalis and its humans and host. And I'm going to particularly focus on the role of parasite extracellular vesicles, EVs, and the host neutrophils in infection. An introduction to this parasite, I've already said it's sexually transmitted. It's a human parasite. It's a flagellate, a single-celled eukaryote that's flagellated at the anterior end, quite highly flagellated, as you can see in these pictures here. It's a very highly divergent eukaryote. So it's most closely related of the other parasites that we all study. It's most closely related to Giardia. It's microaerophilic, which is to say it doesn't really need much oxygen to survive. And it resides in the urogenital tract and is exclusively a human parasite. Now, trichomoniasis is really a very widespread and very prevalent disease, the infection caused by this parasite, with about 250 million cases being reported throughout the world each year, with 3.7 billion in the US. So it's very prevalent. A study done some time ago by the CDC, they showed that randomly going into 6 different inner city areas in the US, about 3.1% of US women of reproductive age showed that, evidence of having been positive for trichomonas during their life. And both men and women are infected as a sexually transmitted infection. However, most men are asymptomatic. Up to 95% of men are asymptomatic, and only probably about 50% of women show symptoms. So this is one of the reasons, even though it's so prevalent, few people know about it. Still, with 50% of women with symptoms and the high rate of infection, there are quite a few symptomatic infections. Most of the times, the symptoms in women are vaginitis or cervicitis, and in men, urethritis. What I'm showing you here in this pie chart is that if you look at the number of syphilis, gonorrhea, and chlamydia cases reported annually in the US, they come nowhere near to the number of reported trichomoniasis cases. So indeed, very prevalent. It's the 3rd most common STI in the US, third only to human papillomavirus and herpes virus. There are other problems associated with trichomoniasis, pregnancy-related problems particularly. Infection is associated with preterm delivery, which of course results in low birth rate and increased infant mortality. There's some long-term effects, this particularly in untreated infections, which happens when they're asymptomatic. Being associated with infertility in both men and women. The long-term infections are associated with increased HIV transmission, and there's also an increased risk of aggressive prostate cancer and cervical cancer. What I'm showing you here is an infected cervix as opposed to an uninfected cervix. Now, this parasite resides throughout the urogenital tract, but it really has a preference for colonizing the cervix. And what you may be able to see, hopefully can see here, is that when the cervix is colonized, it changes quite a bit, and you see those punctate spots there. What those punctate spots are actually lesions. So this is where the parasite is binding and lysing host material, lysing the epithelial layer there of the cervix. So it causes inflammation when this happens. It's an extracellular parasite that adheres to the surface of the host cell, as I was sort of showing you some of the end results of that adherence, and lyses when it's there, when it's lytic. And it has to do this to establish and maintain infection. So this is very unlike, its extracellular property is unlike some of the other parasites you're going to hear from in the session. Some forms of leishmania that Steve will talk about are intracellular. The malaria parasite that Mimi will talk about, it's completely an obligate intracellular parasite. But trichomonas is an extracellular parasite. So it's very important, this interaction with the host cell and binding to the host cell is very important for establishing and maintaining infection, yet we really don't know a whole lot about the details of how this is going on. We know that there's an abundant lipoglycan that mediates the parasite-host interactions. Other groups have shown extracellular cysteine proteases are important. They're partly involved in this lytic process. Parasite surface proteins, many different ones have been implicated in adherence, as well as secreted proteins. And more recently, we've worked on a lot of these things over the years, but more recently, my team has started looking at whether or not T. vaginalis secreted vesicles might be involved in this host-parasite interaction. We didn't really set out to study these secreted vesicles. What led us to start studying extracellular vesicles in Trichomonas was finding that there were several tetraspanins, these proteins called here tetraspanins. They span the membrane 4 times, and hence the name. They act as outside-in surface molecules for signaling the environment from outside to the inside. And we found that several of these were in the surface proteome of T. vaginalis. At that time, tetraspanins had really only been studied in animals, and there's 33 in mammals, but they've been found to be involved in a lot of different processes: cellular proliferation, adhesion, migration, fusion, etc. And we were intrigued by this because these were processes that maybe the parasite really needs to be doing to establish the infection. Also, these proteins aren't even found in most unicellular eukaryotes. When we first found them, and I think still it's only in Trichomonas and Antamoeba that you see tetraspanins. So we thought that was intriguing. And the thing that really caused us to focus in on them is this shown here. Oh, sorry, just a second, let me go back. Okay, when the parasite is not in the presence of the host cell, so when we just have the parasites separate from the host cells, this protein is on the surface, as you hopefully can see here. That's how we found it in the surface proteome. But when you put the parasites in together with the host cell, you suddenly see this accumulation of all this tetraspanin inside the cell in these really huge complexes, vesicle type complexes. Before I go on, I'll say the first part of the work I tell you about the EVs are, about tetraspanin and EVs are going to be, that was done by Olivia too, a very talented graduate student in the lab. So that caused us to really say, well, what is going on? What are tetraspanins in the first place? And we went into the literature and found out that, in fact, tetraspanins are the membrane markers for certain types of organelles called exosomes, or I'm also going to refer to them as EVs here. So you're going to hear and see on the slides a mixture of exosome EVs. Just consider them the same for this talk. Excuse me. So what we learned was that exosomes are these secreted vesicles that are made through these multivesicular endosomes, and we think that's what we were seeing was a real ramp up of the multivesicular endosomes inside the cell when the parasite contacts host cell, and that they're marked by these tetraspanins. So that led us to say, what are exosomes, and would we maybe be interested in looking further at them? Because at this time, they had really only been studied in mammalian cells. When we first started, a few parasite groups sort of came on these vesicles at the same time, but none had been published yet. We all published about the same time. So what are they? Okay, as you see in this cartoon, this animation to the left, exosomes deliver cargo from one cell to the other, and in doing so, they're important as intercellular communication. So what you're seeing here is there's a source cell that secretes the exosome. The exosome is taken up by a target cell, and in doing this, they interact, they talk, they exchange cargo from one cell to the other. Having been highly studied in mammalian systems, they've been shown to play roles in cancer, immunology, and neurobiology. And aside from just the interesting biology, they're really a big emerging frontiers where EVs are being now developed to deliver drugs and vaccines to specific targets. And they're also being utilized as biomarkers for certain disease, because certain disease states of a cell will have EVs with certain markers that aren't in a non-disease cell. And so there are ways to very quickly diagnose for disease. So our next question was, does T. vaginalis even make EVs and secrete them? And we found they did. I'm just showing you some of the data from a publication a while back, where Olivia is looking at her isolated EVs, and you see the picture here, the electron micrographs here. They look quite similar to what had been described for human EVs. Sizing of them showed that they had a mean size of about 95 nanometers, which is in line with the size of exosomes. And then the proteome, the first proteome we did of a T. vaginalis EV, what I'm showing you here are the red stars are proteins that have been described to be almost always present in an EV from any tissue type, from a mammalian cell. And as you can see, pretty much all of the main conserved players from mammalian EVs are in our TV EVs. So we thought we would like to know whether or not these T. vaginalis EVs can actually be secreted from the parasite, delivered to the host cell, and modulate host cell metabolism, and also mediate interspecies communication, the intraspecies communication that is happening in mammalian cells. And one of the first things we did was to look at the effect of EVs on vaginal epithelial cells, referred to as VECs here, on upregulating two cytokines that we already knew. We had already shown the parasite could upregulate in host cells, and that is IL6 and IL8. And the experiment that you're looking at here, the host cells were incubated with either the negative control BSA, parasites, or EVs, and we tried to use the amount of EVs we thought would be also delivered by the same amount of parasites that we put into these experiments, and let incubate for a while, collect them, and measure IL6 and IL8. And what you can see is indeed exosomes alone or EVs alone induce a strong IL6 response, as well as an IL8 response. In the next experiment, what we did was we tried to prime. Or we did prime. So what we did is we treated those host cells first with either our negative controls, PBS, BSA, or the exosomes or the EVs, waited for a while, washed everything away, and then added parasites to the host cells and looked. And what we found is that with IL6 priming did not really change the amount of IL6 that was produced, but priming greatly reduced the IL8. And we thought this was very interesting because IL8 is one of the major cytokines for recruiting neutrophils to the site of infection. And it was known that there was lots of neutrophils recruited to the site of infection. This is showing you an infected cervix here, where there's about 100, if you take that pus, there's about 100 neutrophils to every parasite. And so we thought that this might indicate that the exosomes are dampening the IL8 response, and this might allow the parasite to combat the parasite clearance that would happen by neutrophils. We also wanted to know if they could affect parasite attachment to host cells. What you need to appreciate here is that the different clinical isolates of T. vaginalis vary incredibly much in their ability to both bind and lyse host cells. And so what you're seeing here is 6 different variants. The ones to the left we're calling poorly adherent, to the right highly adherent. These are, I'm sorry, not variants, of clinical isolates of T. vaginalis. So what we did is we asked does the exosomes from the highly adherent parasites increase the adherence of a very lowly adherent strain, the G3 strain, and is that different? So the exosomes from poorly adherent parasites would not increase as much. And so in the next slide, you see that is in fact what we see. We always use G3 as the adherent parasite, and then treated with here G3 exosomes, T1 exosomes, the different strains. These are the lowly adherent. You don't see any increase whether you treat the parasite, the VEC, or both. But if you treat with exosomes from the highly adherent strains, in fact, you do see a very statistically significant increase in the ability of this lowly adherent parasite to bind host cells. So from this work, I think we established that the EVs do deliver cargo to host cells. They elicit immune responses that increase the parasite adherence to host cell. This is done in a strain-specific manner. So we think the likely roles in infection is to assist in colonization by increasing parasite adherence to host cells and by combating clearance by neutrophils by downregulating IL8. So now to tell you some more recent work done by a talented postdoc, Anand Ray, who joined the lab. He's a well-trained biochemist, and so he really wanted to know what are the factors, both on the host cell side and the parasite side, that mediate this uptake of parasite EVs by host cells. So Anand devised or modified a system that had been described from mammalian systems, where he labels the EVs or exosomes with R18, which is a dye. And when it's labeled on the surface of the vesicles and very dense, it's self-quenching, so it doesn't fluoresce. But then when you have an uptake by the host cells, sorry, it's a very overly sensitive cursor here, uptake of the EVs by the host cell, that diffuses the dye and it's highly fluoresces. So we use a FACS based assay to monitor uptake. So this is just the essence. If you don't have any host cells, you get no fluorescence. This is with EVs, and this is a negative control type of organelle from the parasite that even when it's labeled, it's not taken up, showing that this uptake is fairly specific for a specific type of vesicles. It's time dependent and temperature dependent, not surprising, but this just showed us that this is an active process. It's pretty much under our conditions. It levels out about 30 minutes. So all the experiments I'll show you were done at 30 minutes incubation. Well, an interesting thing that Anand also observed, which hasn't been published actually, is that that EVs that are grown at lower pH are much more efficiently internalized by the host cells. And that's relevant because the vaginal pH of the pH of the vaginal tract is less than 5 pH. pH 5. We typically grow our parasite and do these experiments at pH 6.2. But if you actually grow the parasites at 5, as shown here, and then look at the effects of growing the exosomes that are produced at physiological pH, you get a much greater uptake, which is indicating that uptake is favored by physiological pH, which makes us believe a lot of this is probably happening in the human. Okay, then on and went to see what type of surface molecules on the host cell might be involved in uptake. And he looked at a number of things. I'll tell you what he found that looked involved. These were glycosaminoglycans, or GAGs. So GAGs are on surface proteins. They're carbohydrates on vaginal epithelial cells. I really apologize for this. I don't know why it's so sensitive. On vaginal epithelial cells, the main GAGs are composed of heparan sulfate or chondroitin sulfate. So he looked at, we got wild type CHO cells from Jeff Esco at UCSD, but we also got mutants in these particular GAGs. So he looked at the wild type mutant that had no heparan sulfate, and then mutants that had no GAGs at all, and asked about uptake. And as you can see in this next slide, this is the wild type. The uptake is quite good. But if you lack the heparan sulfate, it's highly reduced, and lacking all even reduced more. And these are other experiments. This is a competition experiment. This is taking the heparan sulfate off enzymatically. All these 3 different approaches very clearly indicated heparan sulfate to be involved. This GAGs of having heparan sulfates to be involved in the uptake of the parasite exosomes by host cells. So then we thought, well, let's look at our surface proteomes. We have surface proteomes from a bunch of different EVs from a bunch of different strains. So we said, let's look at the surface proteomes and let's try to figure out if we can see anything that looks like it might be carbohydrate binding. And interestingly, the most abundant protein in the membrane of the EV, this proteins we're calling TV4-AGT, we found that it would be predicted to bind carbohydrates by these phyro2 type things. So then we set out to ask, well, is it binding heparan sulfate? And as you can see here from these ITC plots, in fact, it does bind, shown here when you're trying to bind this protein to heparan sulfate and measure the binding as opposed to the negative control BSA. So we show that it's got a pretty high affinity, in fact, for heparan sulfate. And then another approach, we did competition experiments. This is the uptake of the EVs. If there's no protein, this is if the protein is nonspecific, BSA, and this is the knockdown when you compete with this ligand on the surface of the EV. So we're quite sure this is one of the major players on the surface of the exosome that initiates uptake by host cells. Another interesting thing that also unpublished that we haven't that we observed is that the uptake of EVs from highly adherent strains, shown here in these two, is much more efficient than lowly adherent strains. So this is consistent with a role for EVs and parasite adherence. But also when we quantify the amount of the 4-AGT in the EV proteomics from these strains, we find we see a much higher amount of this ligand in the highly adherent strains than the lowly adherent strains. So what we've been working on lately that we're not quite ready to talk about is cross-linking this ligand, this exosomal ligand, to host cells to try to identify the host cell receptors that are involved in uptake. How about the pathways used for uptake? This just reviews things that most of you know. There are two major endocytic pathways, the clathrin-mediated and the caveolin lipid RAF-mediated endocytosis. So Anand used inhibitors first of the clathrin-mediated endocytosis pathway and showed that there was very little inhibition of uptake. However, when he used different types of inhibitors that inhibit different steps of the caveolin-dependent endocytosis RAF-mediated pathway, he saw quite strong inhibition of uptake, as shown here. As you guys probably know, RAFs are full of cholesterol. If he depletes cholesterol, as shown here, it also inhibits the uptake. If you restore the cholesterol in the membranes, you partially restore the uptake. And a final thing that indicated to us that this was that the uptake was through caveolin lipid RAF-mediated endocytosis. Are there certain cell types that just don't have much caveolin in them or much lipid RAFs? And one of them is a very common cell called HEC293. And so we hypothesized that if this is really true, what we saw from the inhibitor studies was really true, that we should get much less uptake from HEC293 than we do from our other cell lines that we had been studying. And sure enough, you see that's true here. And if we transfect in caveolin 1 into the HEC, then we increase the uptake. So for this portion of the talk, I just want to summarize with that we find that the host cells do internalize these EVs. Proteoglycans are on the host side are mediating the uptake. We found the first ligand for EVs, this ligand that binds heparan sulfate on GAGs that are on the parasite side, and that they're internalized via caveolin lipid RAF dependent endocytosis. So since this is a symposium on molecular parasitology, I'd be remiss not to say that in fact, over the years now, EVs have been shown to mediate many different host-parasite interactions in many different parasites, including Leishmania, T. brucei, malaria, and the parasitic worms, etc. So this is an area of pretty intense research in parasitology now. I'm going to switch gears and the remaining bit of time I have, and I want to talk about something that's much more host specific, and that is the immune response to T. vaginalis. It's very poorly characterized, unfortunately. This is a rather understudied parasite. And there are no good animal models for studying it. And that's, of course, you can't really study, or you can, but it's harder to study without an animal model. And the worst, what even worse, is that if the animal models that we have been able to, we and others have mostly have been able to establish, you have to immunosuppress the mouse or the gerbil to get infection. And of course, can't really study the immune response when you immunosuppress. Um, we do know that neutrophils are recruited to the site. I showed you this picture already before, and very highly recruited to the site of infection. They're very destructive cells, neutrophils are. So they're very likely contributing to this inflammation associated with pathology. And you would assume that since they're recruited to the site of infection and they're known to be killer cells, that they would be able to kill the parasite. But when we first started looking at this, we started scanning through the literature, and no one really had ever gone forward and actually shown that. It was kind of being assumed, but it hadn't been shown. So an incredibly great postdoc, Francie Mercer, who's gone on and has set up her own lab now to continue these studies. Um, what Francie did was she just asked, okay, let me set up a system. Let me see, can we directly show that human neutrophils do kill T. vaginalis in vitro? So she is working with donor neutrophils gotten from human donor blood. And what she did here, and most of the experiments that I'm going to show you, this is sort of more or less the setup of what she does. She labels the parasites and the neutrophils with different fluorescent dyes, as indicated by the red and the green here. Then she adds human serum, and I should say human serum has to be added, or this she doesn't see any killing at all. And when she adds the human serum, she then co-cultures them for various lengths of time. Let's just say 2 hours is the usual experiment. And then she counts the survivors, counts the parasites that have survived by flow. When she first set up these experiments, she also looked to see if the parasites were killing the neutrophils, and they were not. But she does see killing of the parasites by the neutrophils, and that is shown here. This shows you even with a 1-to-1 ratio of neutrophil and parasite, you get some killing, about 4 to 35% or so in this assay. And if you increase up to 16 neutrophils per parasite, you can see under these conditions 100% killing of the parasites. And of course, this is far, far less than that 100-to-1 ratio that I was telling you about before. So we think they're probably quite good at killing these parasites. And what she's done for the rest of her experiments is to be in the dynamic range of the experiments of the assay. She's usually done a 4-to-1 ratio of neutrophils to parasites. Is this donor specific? That was important to ask. And the question is, the answer is no. This is 20 different donors, and this is using that 4-to-1 ratio. You see this, this is more or less, the killing is not that variable between donors. Well, how do neutrophils kill pathogens in general? Probably a lot of you know this, but I'll quickly review it. There's 3 different well-described mechanisms. Phagocytosis, so this is when they just do whole pathogen engulfment. There's degranulation, and this is when they exocytose toxin-containing vesicles that then fuse with the pathogen and kill it. And then there's this other mechanism called netosis, which I think is so incredibly fascinating, where the neutrophil basically casts out its nuclear contents. And sort of this snotty web from the chromatin and the snotty DNA that it casts out, it engulfs and snarls the pathogen, and then typically you also get degranulation that ultimately kill this pathogen. Okay, so we of course wanted to know, are they using these mechanisms? This has been published, and I won't go through it for the sake of time, but Francie did, I think, quite convincing experiments coming at it from a number of different angles, which eliminated the killing to be by netosis or degranulation. So then how about when we looked at inhibitors, classic inhibitors of phagocytosis, cytochalasin D and ortvaninin? Would this Would this indicate that we need contact-dependent phagocytosis for killing? And as you see in these data, the answer were yes, that these cytochalasin D and wortmannin were very, very efficient in inhibiting parasite death at all these different MOIs, the different ratios of neutrophils to parasites. So we're looking at all these data, and some of it didn't make sense. First of all, when we looked at our FACS plots, we did not see a clear population shift. And this was always the case, and that was kind of inconsistent with a whole phagocytosing of the intact parasite. The other thing that intrigued us is that most pathogens that are phagocytosed by neutrophils or other immune cells are quite small. Okay, so sort of this is one of the classic bacillus. It's very, very, very small, so pretty easy to phagocytose, something that small. Well, a neutrophil, it's just the multi-lobe nucleus of a neutrophil you see here. A neutrophil has an average diameter of 8.85 microns, excuse me, whereas T. vaginalis is a pretty good sized cell with about 5 to 15 micron average diameter. So this caused us to say, is it really phagocytosis? Should we look more carefully at this? And luckily we were able to do that. Actually only a few years, maybe 5 years before we were doing these experiments, had this new technology called imaging flow cytometry become, relatively standard at American universities. And so we were able to do this imaging flow cytometry to look further at this uptake. What is an imaging flow? You probably know, but if not, it combines the quantitation of flow cytometry with high resolution microscopy. So basically you can visualize and do signal quantification at a single cell level. So we did this, Francie did these experiments. And here she has the parasites labeled red. The neutrophils are labeled green. And now here's the cold culturing. And of course, we did this hundreds of different times, but I'm just showing you a snapshot picture. What you can see is that the parasites are being taken up by these neutrophils, as you see in the merge here. But they're appearing as small fragments or bites, you might say. So now this could be that they phagocytosed them whole and then were breaking them up once they were inside the neutrophil. That's certainly could happen. So then we took advantage of something we knew about the parasite. We can kill them by heat killing them. They're dead. They don't make ATP. They can't really control their anything in their cell, really. But they're intact. And when we feed the neutrophils these dead intact parasites, we don't see these bites, as you can see here. You just see whole cell uptake. So this indicates that the parasite fragmentation that we saw with the live parasites, that the fragmentation was occurring before death. And this became very reminiscent about at this point. And in fact, I will give Bill Petrie and Katie Ralston credit here. I'm not so sure we would have gone off to look at this phenomenon if we hadn't known about their work, which I'll get to in this slide in a second. But their work really revived and started a new area where of study of cells biting other cells to kill them, and it's called Trobocytosis. Trobocytosis is one cell nibbling at the adjacent cell, taking bites, and not always, but in some cases it results in cell death of the cell that's being bitten. So we asked, were these neutrophils killing T. vaginalis by trobocytosis? This phenomenon was described many years ago in immune cells, where trobocytosis was used to exchange membranes of membrane proteins, so antigen sharing between immune cells. But Katie Ralston in Bill Petrie's lab, our next speaker, actually showed something very interesting, published it in Nature in 2014, that the parasite that Bill's going to talk about, E. histolytica, actually kills host cells through trobocytosis. And in this cartoon, basically what you're seeing is the parasite taking increasing number of bites from the host cell, ultimately damaging the membrane to such a point that there's host cell death. Now, what we would be seeing would be the opposite. It would be the host cell biting the parasite and killing the parasite that way. And since there's been actually a number of studies done and shown that there's a number of different ways in which trobocytosis is used. Intracellular bacteria can be exchanged between macrophages using trobocytosis. While we were doing our work, neutrophils were shown to trobocytose cancer cells. But trobocytosis hadn't been observed for an immune cell to kill a pathogen, so we wanted to really nail this down. We did this by doing live cell spinning discs, confocal discs, spinning discs confocal. This is just showing we label the parasite green. We don't label the neutrophils. We put in propydian iodide at the end because this shows us when the cell has died. This is just showing you bright fields or no bright fields where you don't see the neutrophils. So these are neutrophils, these are the parasite. And you can see that the bites are being taken, and the bites are, what's most important is the bites are taken, precede death. So this is an active process by which the neutrophil is killing the parasite. It has to do a lot of biting before you get death. And we did the average number of neutrophils involved, bites before death, et cetera. We did all that sort of housekeeping. What this is just showing you some 3D super resolution confocal, where again you can see nice images. I would show you a video, but it's I think too much bandwidth. But you can see the parasite here is in green. These are the neutrophils swarming around and trapping the parasite, and you can see bites of the parasite going in. So what is the mechanism of killing? We know a little bit about this. The serum and FC receptors are required for efficient killing. I won't show you that data, it's published. How does the nibbling occur? These neutrophils are chock full of serine proteases. So that caused us to think probably serine proteases might be involved. So we use this broad-based serine protease inhibitor, AEBSF. Showing you here, this is just the control that you've seen before. You get a lot of biting. You just see this green stuff from the parasite going into the neutrophils here. Or if you use the inhibitor, you don't see that, and this is just quantified. The number of bites is really far down, the number and the killing is really reduced. So the host serine proteases from neutrophils are required for this killing mechanism of neutrophils. One last thing, this is all unpublished and what we've been working on now, but the pandemic has really slowed us down. We can't get blood, donors don't come to campus. But this is quite interesting. It turns out that clinical isolates of T. vaginalis vary quite a lot in whether or not they're susceptible to killing by trobocytosis in this 2-hour assay. So all the data I've been showing you has been on this G3 parasite here. So it's very sensitive. So you get very high level at this, under these conditions, very high level of parasite death, okay? We have many other strains that are also very sensitive, so killed very well by trobocytosis. But we have clinical isolates that are very resistant and aren't killed. So we've been trying to figure out what's the difference between sensitive and resistant parasites. This might have something to do with, might tell you something about whether you're symptomatic or non-symptomatic or whatever, may have a lot to do with what happens physiologically in infection. The simplest thing was we thought they wouldn't swarm. So we thought that the neutrophils would not swarm the parasite, and that the parasite would be, the strains that are resistant would be secreting out something that kept them away or whatever. That's not true. They swarm just as well, as you can see here. Here's the 2 resistant strains. Here's the 2 sensitive strains shown here. Sorry. They also bite. This is the number of bites that they take. Doesn't really differ between trobocytosis sensitive and resistant parasites. So we have a number of other hypotheses that we're trying to work on now to see how it is that resistant parasites are resistant, because I think that's quite interesting. summarize and I'll wrap up. Neutrophils, but this section on neutrophil killing, they can very quickly and efficiently kill this parasite. In vitro, the killing is strictly contact dependent. I didn't actually show that data, but it's published. They kill by biting. The parasite to the point at which the trobocytosis or nibbling kills the parasite. Serine proteases and antibody Fc receptors are required for this mechanism of trobocytosis, killing of pathogens by neutrophils. Through this work, we described a new mechanism for killing of a pathogen by a neutrophil. Then we think maybe they use this really when they're attacking very large and highly motile extracellular organisms, which is certainly what Trichomonas is. And then the thing that we're really pursuing now is that not all strains are killed by trobocytosis, and why is that? So I'll wrap up now and be happy to take questions. Oh, maybe I should say quickly, I acknowledged Livia and Anna and Francie, who were the main leaders of the work I've talked about today. But there were, of course, other contributing lab members who helped out a lot, and they're listed here. And funding from the National Institutes of Health. Thank you.
Am I live? I don't know yet. Okay.
I can hear you, Steve.
Okay. Please ask questions or into the chat box. I'm still learning how, the question box, I'm still learning how to do this thing, so I might need some guidance. While we're waiting for that, Pat, I have a question. There was a very interesting talk by Steve Barkey just before yours in another session, saying that neutrophils that are isolated are really different from neutrophils in situ, largely because of downregulation of a lot of pathways by sialylated glycoproteins and things like that. And so so I'm wondering whether that intersects with what you reported about serum or FC requirements. But my other question is about the animal models for these sorts of things. Is any, a lot of people have speculated that dysbiosis of the vaginal microflora is required. And I'm just sort of curious about both of those things.
Yeah, they're very good questions, Steve. Truly everything you say, both of the influences of the role of neutrophils and how they kill, they're very good points. Not much has been done because we don't really have the animal models to look for the dysbiosis of. As far as the neutrophils isolated as opposed to in the animal, again, without the animal models, I think we have very little concrete that we can say, other than all of those are really good things to keep in mind in terms of caveats on the conclusions. We have tried and are trying to get an animal model established, and we took the route of looking at a prostate model because there's so much problem with the vaginal tract, partly of what you said with the different the different organisms, the microbiota and how it varies and so forth. So we thought if we went into a prostate, we might have a little more controlled environment. And we do have the model established. We haven't published it yet, but that would be something that we would hope to be looking at in the future if we can get a robust model.
Good.